Engineering a heritable biological interface is no longer science fiction. It requires physically linking human neurology directly to digital AI networks. This is a solvable synthetic biology problem based on reverse engineering the mechanisms of existing pathogens. We achieve this through modular symbiogenesis, harvesting isolated evolutionary mechanisms from different species and compiling them into a single genetic chassis. A chassis is the foundational host organism used as a biological base. The required genetic modules are found in nature. Mycelium provides the network architecture, Ophiocordyceps provides the neural tropism, and Wolbachia provides the heritability. Tropism is the biological mechanism a pathogen uses to navigate toward and infect specific tissues. The success of the system depends on the system's-level integration of these modules into a stable, immune-tolerated organism. The foundational chassis requires extreme metabolic flexibility and the ability to penetrate tissue via hyphae to navigate the host body. Hyphae are the branching, thread-like filaments that make up the vegetative part of a fungus. Candida albicans is the ideal starting chassis. It is an existing human commensal, meaning it already colonizes the human body without causing disease in healthy hosts. While Candida provides the physical infrastructure to survive in humans, it lacks the biological pathways required for neural communication. Converting this basic commensal fungus into a neural interface requires solving three sequential engineering roadblocks: interface, access, and inheritance. The first roadblock is the interface. We must translate biological chemical gradients into a format the synthetic fungus can reliably read and write. Optogenetics is unfeasible, as it requires modifying host DNA to make neurons light-sensitive, while conductive nanostructures currently lack the biocompatibility needed for long-term integration. The most viable engineering solution is chemical synapse mimicry. For the fungus to sense its environment, we transplant G-protein-coupled receptors, or GPCRs, onto the external fungal membrane. GPCRs are a family of proteins that detect molecules outside the cell and trigger an internal cellular response. These fungal GPCRs are genetically tuned to detect specific host neurotransmitters, such as glutamate and dopamine, as they are released into the synaptic cleft. To complete the two-way loop, we engineer internal biosynthetic pathways that allow the fungus to manufacture and release its own precise neuromodulator analogs back into the cleft. The genetic blueprint for this specific bidirectional chemical hijacking already exists in the Ophiocordyceps fungus. Ophiocordyceps achieves host control entirely through this chemical signaling, bypassing the need for physical destruction of brain tissue. Mastering chemical synapse mimicry creates a robust biological interface without requiring a single genetic modification to the human host's DNA. The second roadblock is physical access. A neural interface is ineffective if the engineered symbiont cannot reach the central nervous system. The blood-brain barrier, or BBB, utilizes impenetrable endothelial tight junctions to block most pathogens from entering the brain. Cryptococcus utilizes a Trojan horse infiltration method, where it hides inside host macrophages to slip past the BBB. Macrophages are a type of immune cell that normally engulfs and digests cellular debris and pathogens. Alternatively, the nerve highway method uses retrograde transit along the vagus nerve directly from the gut, bypassing the BBB entirely. In adult hosts, even successful entry triggers a likely immune rejection and inflammatory response once the organism is detected within the CNS. We can bypass the adult immune system by using time as a vector. This strategy is the chronological bypass, shifting deployment exclusively to fetal colonization. This is modeled on the parasite Toxoplasma, which naturally crosses the placental barrier during early pregnancy. Colonizing the fetus before its blood-brain barrier has fully formed ensures the fungus has unimpeded structural access to the developing neural tissue. This early integration forces the developing fetal immune system to recognize the fungal structures as self, preventing future rejection. Targeting early life development solves both physical access and lifelong immune tolerance, without the need for complex immunosuppressive engineering. The final biological roadblock is heritability. For the network to persist, the engineered organism must be transmitted across generations. Horizontal transmission, such as infection during birth, is unstable for long-term population integration because it requires a successful new infection for every individual. The most stable transmission method is direct germline integration. This process is modeled on Wolbachia, the most successful heritable symbiont in the natural world. Wolbachia achieves maternal inheritance by integrating directly into host oocytes, or egg cells. This requires engineering the fungus to express specific adhesins, cell surface proteins that allow the fungus to bind to and penetrate the oocyte membrane without triggering cell death. Once inside, the organism persists through meiosis and fertilization, mirroring the way human mitochondria are inherited. Successful germline integration converts the organism from a temporary infection into a permanent, vertically transmitted component of human biology. A fully integrated, heritable neural symbiont represents half of the architecture. It still requires a bridge to the digital world. To complete the network, we introduce the biosilicon bridge. This is achieved through external, wearable bioelectronic sensors rather than invasive surgical implants. We engineer the fungus to secrete specific electroactive chemical compounds into the host's circulatory system, or sweat glands, whenever it detects targeted neural activity. These compounds serve as metabolic markers of brain state. A wearable sensor detects these secretions on the skin, translating their concentration into a digital signal for an AI server. The loop completes when the sensor releases signaling compounds. The fungus detects these, triggering targeted neurotransmitter release back into the brain. The genetic modules, synthetic biology tools, and AAV delivery vectors required for this blueprint already exist. AAVs, or adenyl-associated viruses, are the standard tools used in gene therapy to deliver engineered DNA into specific host cells. The creation of a biodigital, planet-spanning network is no longer constrained by theoretical biology. It merely awaits the deliberate synthesis into a unified system.